I wanted to introduce, this is not normally usual, but since we have a new theory about cancer. Until now, the cancers have been considered organ-specific. You have an organ in the liver, you have cancer in the liver, you have cancer in the brain, you have cancer in pancreas, and then the medication and drugs have been cancer and organ specific. Now, the new theory, it is based on critical analysis of many publications, about 150 or something. New theory is that cancer are categorized in 3 groups and then can be targeted to be cured independent of the organs. This is completely a new theory and new target, and because of this importance of the subject, we chose that David Joseph present this new theory, which is important, very important actually, and it is very specific for any cancer in the body, independent of the organ. That what means non-organ specific. So it is beyond organic specific oncology, a unified therapeutic theory for treatment of cancer via master regulation of the universal apoptosis network. David Joseph, please, again on stage. We need the he's coming. So the cancers are divided in 3 particular groups based on proteins, malfunctioning of a couple of proteins. This characterize a cancer anyway, so the treatment is not anymore organ specific, but can be targeted through a specific protein. Please.
Thank you.
Wait.
Yeah.
Oh, okay.
Thank you. So I will be presenting you what we are currently experimenting on in the second portion of my presentation with regards to cancer, our cancer team with Dr. Nafusa, Dr. Yo. So the key here for this presentation and for my work on cancer, my experiments on cancer classification has traditionally been focused on the organ of origin rather than molecular mechanisms. This organ-focused approach has obscured the universal biochemical principles underlying cancer. Chemotherapy and other umbrella treatments harm healthy tissues and can induce additional malignancies. Most cancer research analyzes single pathways in isolation, ignoring interdependent molecular networks. You have one lab working on phosphatases, you have one lab working on c-Myc, you have one lab working on translational control. Very little labs see these as a whole, work on these simultaneously. This paper constructs a unified apoptosis network comprising 100 interacting pathways and shows our experiments which will follow. The unified therapeutic theory identifies master regulators that apply to all cancers regardless of tissue currently being experimented on in my lab. The analysis reveals that oxidative stress regulators, p53 pathways, and RNA binding proteins form a universal core. This is what I designed. So as you can see, it is massive. Everything in medicine, in biochemistry. In chemistry is essentially a spider web of interconnections. This framework redefines cancer into 3 molecularly distinct but organ-independent types. The cancer type 1, lack of p53 or p14 ARF. Cancer type 2, DINO malfunction. Cancer type 3, MDM2 overproduction. The human antigen R, a protein I studied extensively, contains 3 RNA recognition motifs, allowing it to stabilize mRNAs critical for cell survival. This is how HER, for short, functions. So, in the nucleus, HER binds AU-rich element containing messenger RNAs, which I previously discussed. R for short. HER then shuttles from the nucleus. It binds to mRNAs and prepares them for cytoplasmic export. HER binds to either APRIL or PP32. They are 2 proteins which serve as adapter proteins for nuclear export. This is HER shuttling from the nucleus. You also have the HER APRIL or HER PP32 complex, which binds the export receptor CRM1. So HER binds to mRNA. HER complexes with the APRIL protein or PP32. And this complex binds to export receptor CRM1. RAN-GTP, the protein you see to your right, enables CRM1 to bind properly to its cargo, the HER protein complex. Without RAN-GTP interaction, the HER complex CRM1 interaction would not be possible. And without this interaction, HER gets stuck in the nucleus. The nucleus in a cell is what contains the DNA. And it has a barrier between it and the rest of the cell. The rest of the cell being the cytoplasm. CRM1 docks on the RIP1P protein at the nuclear pore to unload HER into the cytoplasm. R1P, 1P. Now, HER, the key link to cancer, HER is exported to the cytoplasm in oral cancer cells in a different manner from that of normal cells. This is just one example published in Nature. The HER protein is found extensively in the cytoplasm of many cancers. In fact, most cancers. And whereas in healthy cells, HER is mainly in the nucleus compared to cancer cells. Keep that in mind, it's going to be important later. Under low damage conditions, such as UV light, HER stabilizes 2 proteins, P21 and prothymine alpha. They're mRNAs promoting cell cycle arrest and survival. So you have HER under damage condition, which induces P21 and prot alpha production. So these 2 proteins prevent the cell from dying, a process called apoptosis. Now, HER also increases the production of the SIRT1 protein, which suppresses p53 activity via deacetylation in this complex process. You see right here, HER is in the middle, it's the heart of the spider web, upper right, SIRT1, and then inhibition of p53. By enhancing SIRT1, HER helps cancer cell block p53's apoptotic transcriptional program. This is a complex spider web, but I simplified it for everyone. Everything in blue helps the cell survive. Everything in red helps the cell die when necessary. HER prevents p53 from making the cell die under normal stress conditions, like UV light, for example. This is how the cell dies when HER is absent. you have P53 activating pro-apoptotic proteins called BH3 only proteins. BID, NOXA, BIM, BAD, PUMA. BID gets truncated, and these proteins activate two other proteins called BAC and BAX. BAC and BAX, what they do is they create holes in your mitochondria. What is the mitochondria? It's what converts sugar and oxygen into energy. When these mitochondria have holes, this releases cytochrome C. Cytochrome C then binds to another protein called APAF1. APAF1 forms the apoptosome. The apoptosome is a structure which takes a protein called caspase 9 and binds it to another caspase 9. This makes caspase 9 active. And when caspase 9 is active, it activates other caspases, caspase 3 and 7, and these proteins shred the cell everywhere. So, the apoptosome causes cell death by activating caspase 9. Activation is caused when caspase 9 is organized into dimers. Dimers is essentially duos. HER stabilizes anti-apoptotic mRNAs such as BCL2, MCL1, and SHAP, further inhibiting cell death. You can see this here. You can see inhibitory arrows going to BCL2, MCL1, SHAP. What is SHAP? XAP. It inhibits the caspases I previously mentioned. So, the key here is that you have P53, which kills the cell. P53 is the god of death for cells. HER is its antagonist. It keeps the cell alive. However, this depends on the threshold of damage you are receiving. When damage reaches a certain threshold, HER and P53 go from friend to foe. This is what happens when you have excessive damage. HER fragments into two other elements called CP1 and CP2. CP1 and CP2 fragments increase procaspase 9 stability. Remember, procaspase 9 is what you need to activate in order to kill the cell. So CP1 and CP2 stabilize procaspase 9, enhancing apoptosome formation via activation of the death-inducing caspases 3 and 7. So essentially what's going on is that if you have a certain amount of damage, it's basically a euthanasia process. If you're sick, but you can survive, HER is going to help you. If you're sick beyond the point of no repair, HER is going to euthanize the cell. It's going to fragment. It gets fragmented at aspartate 226 and in the cytoplasm and leads to the death of the cell. So, this paper confirmed that you don't need an apoptosome to cause cell death. If HER produces enough caspase 9, it will lead to the death of the cell. So, the switch allows HER to promote cell death in highly damaged cells while supporting survival in moderately stressed cells. It's a coping mechanism, essentially. The duality of HER P53 interactions is central to designing cancer-specific therapeutic strategies. This is, I looked into the literature at a biochemical level, at the chemistry level, and essentially what I found is that you can separate all cancers into 3 families, irrespective of pancreatic cancer, breast cancer, brain cancer. You have 3 families. This is cancer type 1. which we're currently studying in our lab. So, cancer type 1 occurs when P14ARF or P53 is genetically lost, disabling c-MYC-induced apoptosis. So in cancer cells, you have this protein, c-MYC. c-MYC allows your cells to multiply. However, when your cell needs to be at rest, because if your cells kept multiplying, you would spontaneously combust. So there's a control mechanism on c-MYC. But in some cell, c-MYC goes out of control. So c-MYC going out of control triggers P14ARF, which triggers P53, which leads to a dead cancer cell. However, in cancer type 1, you have either P53, which is dysfunctional, or you have P14ARF, which is dysfunctional. This occurs in 40 to 50% of cancers. You have P53 or P14ARF, which is dysfunctional at a genetic level. So, the optimal therapy here for cancer type 1 is inducing her cleavage to bypass defective P53ARF signaling. This is my hypothesis, which we are currently studying. So, you need, if you have P53 which is off, all the proteins I showed you, BIM, BID, BAX, BAM, all that activation program shuts down and the cell cannot die. But if her gets cleaved, you can bypass that. You can bypass the lack of P53 to kill the cell. So, essentially, some people, because you're going to say, well, Davis, her in cancer goes to the cytoplasm. So why not just keep her in the nucleus? Well, there was actually an interesting study published in December 18, 2024 by a colleague of mine, where she proved that when her is mutated at V225I, a portion of the protein, her cannot shuttle out of the nucleus. It gets stuck in the nucleus. But instead of preventing cancer, it actually induces cancer. Because under situations of extreme stress, her needs to be cleaved. And if you keep it in the nucleus, instead of helping the cell die, it actually helps the cancer cell grow. It produces BCL2 and XIAP. It was a very interesting study published in Cell Death Discovery, December 18, 2024. So my hypothesis is you need her targeting. And remember I told you it's important that in healthy cells, her is in the nucleus, and in sick cells, her is in the cytoplasm. Her is much more accessible in cancer cells and much easier to cleave. If we can harness the potential of cleaving her at asparagine 226, this will specifically target cancer cells and help treat not just cancer type 1, but cancer types 2 and 3, which I will later show. Cancer type 2. Cancer type 2 results from DINO long non-coding RNA promoter hypermethylation, which prevents P53 transcriptional activation. So what do you have here? And it's funny because a 2023 paper proved that these three sicknesses are mutually exclusive. You either have a defective P53 gene or you have a defective long non-coding RNA. You never have both because the end result is the same. P53 cannot cause cell death. So you have these molecules called methyls. They bind to the promoter of the DINO gene. A promoter is what allows a gene to be transformed into a protein. When you have methyls on that promoter, the gene cannot be transcribed. So you do not have DINO long non-coding RNA, and if you do not have long non-coding RNA, P53 cannot function. So the end result is the same. So what I proposed is a target at a genetic level, which we're currently working on. We target DINO promoter P53. Why is this cancer specific? There is no methylation of the DINO promoter in healthy cells. You only find that in cancer cells. Nowhere in healthy cells. So targeting the DINO promoter allows you to target cancer cells without having unintended consequences. This is the problem with chemotherapy. Taxol or cisplatin, they target your cancer, but they target also all the other cells. With a DINO target, you do not target the healthy cells. And finally, you have cancer type 3. So the target therapy for cancer type 2 is demethylating the DINO promoter, which I just said. And now we get to the final cancer type. Cancer type 3 arises from MDM2 gene amplification, which degrades p53 despite an intact gene. So I had an interesting discussion with Dr. Chehanover yesterday. He told me that there are cancers. He told me the MCS7 cell line has a perfectly normal wild-type p53, and he was right. So he's telling me, you have to check that. You have to focus on that in your presentation, and I did. So what's going on in this family is that you have extra copies of MDM2. MDM2 is part of the UPS system, ubiquitin proteasome system. So discovered by Dr. Chehanover. So MDM2, it leads to the degradation of p53. This happens when there's too much p53 in the cell, and it stops p53 from killing a cell unnecessarily. However, in pathological cells, in cancer cells, in 4% of cancers, you have too much copies of the MDM2 gene. And if you have too much copies of MDM2 gene, when the cancer cell is targeted by chemotherapy like cisplatin, for example, the p53 level is supposed to go up to trigger the death of the cell.
But instead of going up, the extra copies of MDM2, the hyperactive MDM2, doesn't allow p53 levels to go up, and the cell does not die. This process occurs in 4% of cancers. That may not seem much, but for a population of 100 million cancer patients, that's 4 million patients. The target here is clear. miR-125B is a microRNA. The Nobel Prize for microRNAs was given last year in 2024. miR-125B inhibits p14ARF and p53. So,
p14ARF is the trigger for cell death.
And it has two connections. The first connection is that p14ARF triggers p53, but it also inhibits MDM2. miR-125B in cancer type 1 actually helps the cell, helps fight the cancer cell, because it also inhibits c-MYC. But when you're in a situation
where MDM2 is out of control, miR-125B is actually detrimental to the cell because it prevents p14ARF from accomplishing its function. So in cancer type 3, p14ARF needs to be hyperactive. It needs to inhibit MDM2 as much as possible. miR-125B does not allow it to do that. It keeps it out of control, but MDM2 is out of control. So our target for cancer type 3 is miR-125B to activate p14ARF, to inhibit MDM2, and to release high levels of p53. So the next steps, we are currently under the experimental phase, is for this theory to apply to neurodegeneration. You have these same proteins in neurons, in brain cells, and they have a massive impact on the longevity of brain cells. So applying everything to cancer cells will allow us to better understand subsequently how to treat neurodegeneration. That is my presentation. Thank you very much.
So next year, probably, we will have the validation of the theory, correct? Thank you again.